So welcome. Again, this is Joanne Geer from the Biopharma Research Council. You are here for a wonderful webinar, and I'm just going to tell you a little bit about our organization. The BRC is a nonprofit association for scientists across all the many silos of biomedical research. So it includes industry, academia, nonprofit, government, and suppliers. And we have a terrific mission, which is to get scientists together to share knowledge and learn about each other. And we do this in order to stimulate the collaborations and partnerships that are needed to accelerate progress. I'm going to share with you a few of our upcoming programs. In July, we're doing a really terrific program on the cybersecurity for connected devices, which includes everything from implants to wearables and even some surgical equipment. Point of Care, that's in Princeton. The Point of Care Diagnostics is a virtual conference that'll happen in August, and in October at Kane University's Institute for Life Science Entrepreneurship Microbiome Update. We have a busy fall. Then we go to North Carolina, October 11th, for the Triangle Biotech Research Symposium, and the focus this year is on data, drugs, diagnostics. In November, back in New Jersey, a really wonderful two-day program on companion diagnostics. And in November, our first ever Regional Pharma Manufacturing Expo. And I'm just going to hold on this for a moment because we're really excited about partnering with organizations and groups that would like to bring content to this day. We have lots of space, so we can give you and your group an interesting place to have an open or closed meeting, whatever you like. And you can get in touch with me in order to learn more about that. And finally, the next webinar in this series on second harmonic generation. So this will be on imaging and quantification of fibrosis in the kidney on September 27th at the same time. You can enter questions through the box on the side of your screen at any time. We'll take all the questions after the presentations. If there's a specific slide you're thinking about, it's great if you put a note there about which slide it is. And here we go. I'm going to introduce briefly our speakers and then we'll hear from them. Dr. Mathieu Petitjean is an experienced leader with 20 years of executive level and international business experience in medical technology and life sciences, including 12 years at General Electric Healthcare in both product and business leadership positions. Dr. Li Shen, formerly from Inclone Systems, Hoffman LaRoche, and Zeus Scientific, is experienced in assay development and validation, demanding regulatory and quality assurance frameworks, related technology transfers, and management of programs with CROs or technology vendors. And there we have it. We're ready to hear from Dr. Petitjean. Dr. Petitjean, if you can hear me, I think you are muted.
Yes, I am muted. Can you hear me now, Joanne?
Yes, we can. Thank you.
Very good. So thank you, everyone, for joining us today, and Joanne again. Thank you to you and to the BRC for inviting us to present. I think we all acknowledge that there is an increasing need both in academia, research and development, and in the clinic to have much more reliable ways to quantify fibrosis. And what we are doing here in Genesis Imaging Services is basically bring a very novel instrument to the market in the US through our partnership with HistoIndex out of Singapore. Second Harmonic Generation is well known as being a technique to be specific and sensitive to collagen imaging. There is a lot of publications available that discuss this method, but HistoIndex has been able to put everything together into a tabletop system that is new to the market in the US, and we are offering pay-per-use services to bring this technology and make sure that everybody becomes comfortable that. Another benefit of the technology is that it has been optimized for pathology-like workflows. So we just need, conventional pathology slides, and I'm going to take you through all the process of both imaging and quantification. First of all, a quick reminder for everybody on the methods that we'll discuss today. Our instruments and the image we create basically overlap two imaging methods. The first one is what we call two-photon autofluorescence, and we're sensitive to different proteins in the cell, and we capture their autofluorescence. Two-photon techniques are particularly interesting. We use red photons, and we combine two photons to create an excitation equivalent to 350 nanometers, which is a blue, highly energetic excitation that can generate autofluorescence of different kinds of proteins. Everything that we're going to show you today comes from NADPH, NADH, as we have optimized the system to be sensitive to those wavelengths. The second optical methods that we use, and this is the one that is sensitive to collagen as optimized in our system, is second harmonic generation. Here, two photons are absorbed and immediately released, and we capture them. The cellular information, the autofluorescence information, shows in red in our images. So this is a way for us to look at cellular structure, tissue structure, but also the overall cellular or mitochondrial health inside the cells. We are not quantitative, we're just qualitative regarding this method, but we see very well the cellular structure. Second harmonic generation will be specific to collagen, more specifically to collagen 1 and collagen 3, and we are sensitive to that in the extracellular matrix environment, and when we combine both, we have these green and red images which basically describe both the collagen structure and the cellular architecture and health. The technique is stain-free. We basically are a non-destructive method, and we return all the samples once they are imaged. And these are the kinds of image we have. I've selected here a liver image where we can compare the pathology image and the image that we generate on our systems. Again, in green would be collagen, so you can see the collagen fibers here, and in red would be the cellular structure, and in particular here, the fat globules. I just want to calibrate everyone on the core of our imaging scale so that there is no confusion on what you're going to see coming down the road. What we see and the images that we're going to show you are captured at 20x. Our resolution is at TX 0.4 by 0.4 micron. We can go smaller at 4EX, for instance, but I want to calibrate the scale of the image you see. So this is here the 20 micron mark, and the fibers here, the collagen fibers that you will see in green, are in the 2 to 3 micron range. So we are not imaging, you know, the collagen triple helix. That's way too small. We are not imaging the collagen fibrils that would be in the 10 to 300 nanometers. We can't see the cross-link, but we can see the effect of all those three parameters into the formation of collagen fibers. So our technique is a method of choice to look at collagen fibers, and also the density of those collagen fibers. The brighter the signal, the more collagen there is for an equivalent surface or cross-section. Now, having said that, what I'd like to show you here is the roadmap of applications we are working on. What we do is basically two things. We do imaging, and then we treat the images to extract parameters related to collagen content and collagen structures. We use machine learning based algorithms, and it takes time to develop tissue-specific and relevant quantification methods. So here is a view of our pipeline. As I said, we can image easily all those tissues, but it takes time to explore a relevant image analysis and quantification tool, develop this tool, and then validate the tool into what we call validation clinical trials. Usually we compare to gold standard using 140 or 200 samples, either in the clinical space or in the preclinical space, and then we become commercial. So right now we are commercial on 3 applications: liver, kidney, and lung. A month ago we presented our applications in liver. We'll present kidney in September, and today's webinar is all about lung. And as you can see, we have bone marrow, we have GI, we have skin, and other tissues that we are currently developing. As I said, 3 commercial applications, and we'll focus today on lung. Before I turn it back to Lee, I would like to calibrate all of you quickly on the image we have. So here is a typical lung, healthy lung image. You recognize the alveolar and the structure. As I said, red is the cellular structure and green is collagen. And let me show you here the collagen image of a healthy tissue. So as you can see, even in healthy tissues, we can recognize very well the collagen. I would like you to make sure that you see my screen. So it's useful to tilt the screen of your laptop to make sure that you see our images the best possible way. So take a second to kind of tilt your screen and make sure that you see the images that we are projecting through the webinar, in particular the collagen images. Now this is a healthy tissue, as I said, and let me show you here a sick tissue. This is a sick lung tissue. Lee will show you in a minute high-resolution images, but as you see, the density of collagen, it's higher, the reticulation and the structure of the collagen is much higher, and you see already some texture degeneration of the cellular structure. Here is the related collagen image, and as you can see, it's extremely rich. We have a lot of information, both in terms of collagen strings, in terms of collagen reticulation, and in terms of texture. Texture encompasses several parameters related to the way those fibers are organized. Specifically to lung, our analysis method follows the following method. The first thing we do, we have an all-tissue image, and we separate 3 regions in the tissue. The structural regions, where we find the structural collagen, we segment that, and then we look at the parenchymal functional areas and the lesional areas. I'll explain how this is done in a minute, but basically we can segment the tissue in those 3 major areas, and of course we can look at how much percentage of functional parenchymal we have versus lesional tissue, for instance. And for each of those regions, and mostly for the functional and lesional areas, we are able to quantify several parameters related to these regions. Well, first of all, the content of collagen. We can look at the morphometrics of the collagen fibers. Here we just mentioned collagen thickness, and we can also look at the texture, the structure of the collagen network. We will show reticulation indexes, for instance. In blue is the functional area, in red is the sick area of the tissue. So here is a little bit more details on how we identify the structural areas. We look at the big collagen objects, we segment them out, and we paint them in green. We just declare those pixels to be green. They are excluded from more specific collagen counts further in the analysis. Now we separate the parenchymal, the functional areas from the lesional areas, through an analysis on texture. This image really shows pretty well, and the corresponding red segmentation shows here. It shows very well that this area is really damaged, and we identify that we've been identified these areas combining textural analysis of the images and also pathology feedback. So we compute the textures of all these areas, and then we ask the pathologist to show us, okay, what would you define as being the lesional area, and then the software understands where to put the boundaries and where to declare that a pixel is either lesional or functional. Also, just as a heads up, we will present data on collagen area ratio, collagen content. So here is after we treat the image, we would count how many green pixels are represented in an area, excluding the structural collagen that shows here in pink. The density comes simply by doing a ratio of the green/collagen pixels over the total area of the region of interest. And just to calibrate everyone here, let me talk a little bit about structure and reticulation. This image shows that we can basically draw an equivalent skeleton of collagen. And the skeleton here is being analyzed in two different ways. We look at the intersections here, we call them nodes or reticulation points, and there is also an interesting parameter which is the distance between two nodes, basically the length of the segment. And once we have this information, there are several parameters that we can derive from the analysis. One of them would be the reticulation index, which is basically measuring the amount of reticulation of the collagen network. High reticulation index correspond to complex collagen networks, and what we do here, we basically count the number of intersections and divide it by the total length of the network. We could also count the number of nodes and divide it by the area of the ROI, but we like this measure. As you can see, that reflects extremely well how the network becomes more and more complex as fibrosis develops. And once we have both collagen content and collagen structure information, we like to plot every data point in what we call the 2D fibrosis chart, which is a very useful way to look at how fibrosis develops in your tissues. Here, usually, we would find healthy tissues. Again, we can measure collagen content and we can compute collagen structure even for very healthy structures. And then somewhere here in this area, we're going to have the very sick tissues where we have a very complex collagen structure, but also very heavy in terms of content collagen structures. And between those two regions, we can see different trajectories, either as you develop in your models, you develop fibrosis, or you cure fibrosis. The trajectories are really different according to either the models that you use or the treatments that we apply. Let me show you here a very, very basic example where we have here two tissue, a control and a bleomycin tissue. We can see the groupings of the control. We can see the bleomycin groupings. I think this was at 14 days, if I remember correctly. We see here a data point where one animal has responded extremely well, if I may say, to the bleomycin, and it's a little bit of an outlier. But we can see here what happens to the grouping and how the collagen develops, both in terms of structure and both in terms of collagen content. And of course, in a minute, Dr. Chen will show you the effect of a compound to reverse or not to reverse the progression of collagen, of fibrosis in your tissues. So that's a kind of quick 360-degree introduction to what we do, and I would like to turn it now to Dr. Li Chen, who will be presenting two very detailed case studies and really show you how we do things and how our data compares to all kind of other data.
Thank you so much, Matt. So today I'll be presenting two case studies. They're both bleomycin-induced lung fibrosis studies in mouse. The case study 1, I'll go through more in depth. Case study 2, more brief, and I'll be using the metric that Matt presented throughout the case study. So the case study 1 is with our collaborator, GenScript. Briefly on the study design, the routes of administration is through intratracheal installation of bleomycin for induction of lung epithelial injury, inflammation, and fibrosis. There's 4 groups of NO4. Group 1 is control, Group 2 is bleomycin, Group 3 is bleomycin plus the drug nintedanib, and Group 4 is bleomycin plus the drug pirfenidone. The study lasts about 21 days, and the drugs are given 7 days after the start of bleomycin. Both nintedanib and pirfenidone, they are marketed drug that has been approved for treatment of idiopathic pulmonary fibrosis. So on the right here, this is a typical slide image of the non-stained slide that we receive for imaging. So now we're going to go into the study images on our cloud-based digital image website. You might have to adjust the angle of the screen to see the red and green images better. So here is a control lung, and if I zoom in, you'll be able to see the alveoli better. So the red color you see here is from 2-photon excitation. They're autofluorescence from the tissue to look at tissue morphology. The green color you see here is from second harmonic generation for collagen. They're the structural bronchial collagen. They line the borders of the lumen of the bronchial tubes. And as you can see here, in control lung, you have the normal alveoli architecture, and the walls of the alveoli is very thin. And this compared to the bleomycin lung. And if I zoom in, you'll be able to see the tissue better. So here in bleomycin lung, you see dramatic changes. You see an increase in the tissue mass. You see loss of the normal alveoli architecture. You also see inflammation, and inflammation is characterized here by this dark shade of gray. So this dark shade of gray indicates to us that there's inflammation and dying cells. In addition, there's also elevated collagen deposition. So if I remove the red channel, you'll be able to see the green collagen better here. So this, in comparison to the control lung, I'm going to remove the red channel. So this light amounts of green collagen is normal in control lung. There's very little here, interstitial collagen, compared to the bleomycin lung. There's a lot of collagen here. And another thing that I wanted to know is that these bright green collagen, the structural collagen, they're actually segmented out when we do a total whole lung collagen quantitation. So now we go into the nintedanib group. So this is bleomycin plus the nintedanib drug group. Okay, so you still see the fibrotic mass, but it's much smaller. You still see pockets of the fibrotic mass, but they're less and they're much smaller. And another thing is there's increase in the alveoli here as compared to bleomycin alone group. Okay, so this is the nintedanib group. And if we look at the pirfenidone group, you see similar results. You still see the fibrotic mass, but they're much smaller and there's pockets of it. You also see increase in the fibrotic, and you also see an increase in the alveoli here. So it looks like there is a drug efficacy with the nintedanib, actually nintedanib and the pirfenidone to reduce the bleomycin-induced tissue injury. So let me go back to the slide deck. So now we're going to regional area segmentation. The left images here is our 2PE SHG images. The signal is push up to the max to help with the regional area segmentation. The image on the right here are the process image for the 3 area. The blue color you see here is the parenchymal area, the functional tissue. The red color is the lesional area, and the green color is the structural bronchial collagen area. So this is a control group. And as you can see, the whole tissue is almost all parenchymal, almost all functional tissue. And you also see a small amount of the lesional area. And the reason why you see this small amount of lesional area here, it's about 4%, is because these control animals, they're not 100% normal, so to speak, because these control group, they're receiving intratracheal installation of saline. So these animals are going through trauma from surgery, and also these lungs are infused with saline. So our technology is sensitive to pick up these minor injury here. So next is these two, the other 2 animals in this group. You see a lot of blue functional tissue. Okay, so now we're going to the bleomycin group. You see dramatic changes. You see a dramatic increase in the red lesional area on the right here, and on the left you see an increase in collagen deposition. And another thing that with these segmentation here, you're able to see the distribution of the lesional mass throughout the whole lung. Okay, and this is the other 2 animals. Here you see a lot of red lesional area. So now we're going to group 3, which is bleomycin plus nintedanib. You see on the right here a reduction in the red lesional area, and then on the left here we see a less of the collagen deposition compared to the bleomycin group alone. And this is the other 2 animals. You see a lot more of the functional blue tissue and a decrease in the collagen deposition. So now we're going to group 4. This is the bleomycin plus pirfenidone. So here you also see a reduction in the red lesional area and a decrease in the collagen deposition. And this is the other 2 animals in the group. So now we're going to the regional area segmentation quantitation graph. We have the lesional area here, parenchymal area here, and structural area here. You see that in control. Okay, so control is in blue. The red is bleomycin. The green is bleomycin plus nintedanib. The purple is bleomycin plus the pirfenidone. In control, you have about 70% functional tissue. 26% of it is the structural bronchial collagen area, and a very small amount lesional area at 4%, and I explained the reason earlier. With in the bleomycin group, you have a reduction in the functional tissue to 36%, an increase in the lesional area to 44%. With the addition of nintedanib and the pirfenidone, you have an increase in the functional tissue to about 60% and a decrease in the lesional area to about 20%. So it looked like these 2 drugs are working to reduce bleomycin-induced tissue injury. Okay, so next, we have data provided by GenScript, who have who uses the regular method. So they provided the H&E and the trichrome stain images and the biochemistry, the hydroxyproline for collagen content, and the Ashcroft score. So if you're interested in comparing our method, our technology with the regular method, you can drop us the email and we're happy to show you the data. But the bottom line is their data correlates very nicely with ours. Okay, so now we go into the images. So these 4 images is a representative image, 1 per each group. It's a very small area within the whole lung. It's about 1 by 1 millimeter. So this is control, bleomycin, bleomycin plus nintedanib, and bleomycin plus pirfenidone. In controls, you see a very nice normal alveoli architecture. In bleomycin lung, you see dramatic changes. You see loss of this normal alveoli architecture, increase in tissue mass, inflammation, which is marked by a dark shade gray here, and elevated collagen deposition. With the addition of nintedanib or the pirfenidone, you see a reduction of this fibrotic mass. It becomes small pockets of it, and also an increase in alveoli compared to bleomycin alone. And if I remove the red channel, you'll be able to see the green collagen much better. Okay, so now we're going to percentage area tissue versus alveoli. The left here is the percentage area tissue. The right here is percentage area alveoli. So in control, you have about 50-50 tissue versus alveoli. With in the bleomycin group, you have a increase in tissue mass to about 68% area, and a decrease in the alveoli to about 32%. With the addition of nintedanib or pirfenidone, you have a decrease in the tissue mass to about 60%, with an increase in alveoli to about 40%. So the significance of quantitative amount of tissue versus alveoli is that the increase in tissue mass, it actually distorts the normal alveoli architecture. And so decreasing the tissue mass is a good news. And for alveoli, as you know, alveoli is involved in gas exchange. So increasing the amount of alveoli is important. Okay. So next we go into collagen quantitation data. So we're measuring total collagen, and this is parenchymal plus the lesional. We exclude the structural collagen. On the left here, you have the percentage collagen area. On the right here, you have the collagen reticulation index. We're measuring for the control, bleomycin plus nintedanib, and bleomycin plus pirfenidone. You see here that bleomycin increases percentage collagen area about 2-fold more than the control, and this increase is reduced with both nintedanib or the pirfenidone. And same thing with reticulation index. Bleomycin increases reticulation index by about 2-fold more than control. And with the addition of the nintedanib or the pirfenidone, it reduces this reticulation index. So reticulation index is a measure of the amount of the complexity of the collagen network. The more the reticulation index, the more complex the collagen network. So if I combine these two metrics together, you have this 2D fibrosis chart. This is group average control, and this is bleomycin group average, and this is nintedanib and pirfenidone. On the y-axis, you have collagen content. The x-axis, you have the reticulation index, or the collagen structure. So with bleomycin, increases not only the collagen content, but also the collagen structure. And with the addition of the both the nintedanib or the pirfenidone, it reduces both the collagen content and also collagen structure. So clearly there's drug efficacy by nintedanib and pirfenidone. So in summary, these two drugs reduce bleomycin-induced tissue injury in terms of the collagen, in terms of the improving the amount, increasing the amount of functional tissue, decreasing the tissue mass, and increasing the amount of alveoli. So now going to case study 2. Case study 2, I'll go very brief in the interest of time. This study is with another sponsor, and the study is also at the level of preclinical research study. Okay, so very briefly about the study design. The route of administration is through aerosol inhalation of bleomycin. The study lasts about 21 days. There's 4 groups of NO4. Group 1 is control. Group 2 is bleomycin. Group 3 is bleomycin plus drug candidate A. Group 4 is bleomycin plus drug candidate B. Drug A is an antibody. Drug B is a small molecule. They both target the growth factor pathway in an effort to treat pulmonary fibrosis. Okay, so here are the images, representative image, one for each group. There's a small area within the large whole lung. They're about 1 by 1 millimeter. So here you have control, bleomycin, bleomycin plus drug A, bleomycin plus drug B. In the control, you see the normal alveoli architecture. With bleomycin, you see a loss of this normal alveoli architecture, increase in tissue mass, you see inflammation, and also elevated collagen deposition. With the addition of drug A, you still have this tissue mass, increase in tissue mass, but you see a decrease in collagen content. With the addition of drug B, you also still see the increase in tissue mass induced by bleomycin, and also the maintenance of the collagen deposition. And if I remove the red channel, you'll be able to see the green collagen better. Okay, so now we're going to regional area segmentation. This is one representative image per group. Okay, so we have the control here. And just to reiterate, the image on the left is our 2PE SHG image. The signal is pushed up to the max to help with area segmentation. The images on the right is the process image, the segment into the 3 area. And the importance of segmenting into the 3 area is that not only will you be able to visualize those 3 areas, but you can quantitate it to compare the amount of functional tissue versus the amount of lesional tissue. In addition, it also helps to segment out the structural bronchial collagen. And in addition, you also be able to see the red lesional distribution throughout the whole lung. Okay, so in control, you see almost all blue functional tissue as expected for control. And this route of administration is through aerosol inhalation of saline. And in the bleomycin group, you see a dramatic change. You see a high increase in the red lesional tissue here, and also elevated collagen deposition compared to control. Okay, next you have, with addition of drug A, you see a slight reduction in the red lesional area, and also a reduction in the collagen deposition induced by bleomycin. And for drug B, addition of drug B reduces the red lesion mass by slightly, and also but it maintains the amount of collagen deposition induced by bleomycin. Okay, so now we're going to quantitation data for the regional area segmentation. You have the lesional, parenchymal, and the structural. For control, you have about 86% functional tissue, 14% is structural. For lesional area, there's little or no lesional area. For bleomycin, you have about a reduction of the functional tissue to about 52%, and an increase in lesional area to about 29%. With the addition of drug A, it increases the functional tissue to 62%, and a decrease in lesional area to about 20%. With the addition of drug B, you have a slight increase in the functional tissue to about 55%, and a slight reduction in the lesional area to about 25%. So it looks like drug A is providing more efficacy than drug B in reducing bleomycin-induced tissue injury. Okay, so now we're going to percentage area tissue versus alveoli. You have the percentage tissue here and percentage area alveoli here. The controls have about 40% tissue and 60% alveoli. In the bleomycin group, you have about increase in tissue mass to 58% area and a decrease in the alveoli to 42%. With addition of drug A or B, you have a slight reduction in the amount of tissue mass to 55% and a slight improvement in the percentage area for the alveoli to about 45%. Okay, so now we're going to collagen quantitation data. You have the collagen area here, and you have the reticulation index here. So here, the bleomycin increases percentage collagen area about 2-fold more than control, and this increase is reduced by drug A to almost the control level. With addition of drug B, the reduction is only very slight. And for reticulation index, the bleomycin increases collagen reticulation index by about 2-fold more than the control. And with the addition of drug A, it provides reduction in this increase in reticulation index. And with additional drug B, it didn't change the reticulation index. Okay, so now combining these two metrics together to see the correlation in this 2D fibrosis chart, you have the control here, and this is group average, and you have the bleomycin treatment here. Drug A is here, and drug B is here. Y-axis is collagen content. X-axis is the reticulation index for the collagen structure. So you see here that with bleomycin induces not only the collagen content, but also the collagen structure. And this increase is actually reduced with drug A and not B. So in summary, it looks like drug A is working better than drug B in reducing the injury induced by bleomycin. And the drug efficacy is in terms of the collagen content and improvement in increasing the amounts of functional tissue. So with this, I'll turn the presentation back to Matt.
Thank you so much, Lee. Before we open up Q&A so everybody can ask questions on what you presented, I just want to close and state a few things that I didn't explain at the very beginning. It talks about, we're going to stay on the same slides, Joanne, if you don't mind. Yes, so Lee's going to share back. Yes, so the elements that I wanted to talk about is our capabilities are mid-throughput, so we can process, let's say for instance, 40 tissues if we put 3 tissues per slide overnight, and the analysis would take a week or 2 weeks depending on the load that we have in our queue. I want to say here that it's a mid-throughput system and we can turn around data extremely quickly. We are also extremely reproducible, and you know, we don't have this well-known intra-operator or intra-pathology reading errors that we are aware of through pathology workflows. The data that I'm talking about has been widely published, and we'll be happy to send you the data that studies desirability aspects. The fact that we don't stain eliminates another source of variability. So we think that we can propose to all of you in R&D, in academia, in preclinical, and in research, a tool that is robust, that we have organized as being very friendly with your existing workflows, very quick. In terms of budget, it does not compare to conventional pathology, but we believe that the benefits of the technology is much higher. Just to close, let me also state that the only thing we need to do a study would be paraffin-embedded tissues, and it's absolutely an option to do retrospective studies if you think that you have a good candidate or if you would like to know more about your studies. So with those last comments, let me turn it back to Joanne, who will facilitate the Q&A session, and I would like to thank you all for your attention over the past 40 minutes.
Well, thank you, Dr. Petitjean. That was terrific, and Dr. Shen. We do have some great questions, and I encourage everyone to feel free to enter your additional questions in the question box. The first question is, when denominator changes, for example, area of lung due to collapse, does percentage area of collagen, is percentage area of collagen a good marker? I guess they're talking about the change if there's less lung surface.
Yes. Lee, I can address that. Yes, that's a good question. We are, as Lee mentioned, we are capable of measuring the area of the alveoli. So we can either use the area of the entire region of interest or subtract the area of the alveoli to really look into the tissue area. And then we would compute the ratio of collagen over the real tissue area. So both options are possible depending on your strategy and study interests.
Beautiful. Our next question, how is your technique different from staining with collagen stains, for example, a Ti-Chrome or Sirius Red, and use a regular image analysis platform, for example, ImageJ?
Lee, would you like to try to answer that?
So our image analysis tools are very sensitive, as you see here. With ImageJ, it's slightly different because our tools are tailored to not just your study, but also has gone through a pathologist review.
Yeah. I think pathology, tissue, first of all, there is variability that we inject in the tissue when we stain, so we avoid staining, and henceforth we don't have to deal with the destructive and variability of what we inject in those tissues. The second, in terms of image analysis and quantification, we are using machine learning algorithms, so it's much more sophisticated than ImageJ, and there are pathologists' interventions that basically teach the software. So I think between staining, non-destructive from tissue processing, and the fact that we use a machine-based learning software would be the two major differentiations between the case that you presented.
Thank you. Our next question. Can distribution of lesional area, for example, central versus peripheral, be quantified?
I believe the quantification of the lesional area is throughout the whole lung, so it wouldn't matter if it's in the center or throughout the whole lung. But we can actually look at a particular area of the lung if you need that. So instead of a whole lung, if it's a particular area that you're interested in, we can also just look at that particular area of the lung.
Okay, well here's someone who wants to get right to the point. They're asking if they'd like to know the price for analyzing each sample, liver, lung, and kidney, and sense of the time, the turnaround time for the analysis. You may wish to address this more on an individual basis, but if you'd like to answer the question, you're welcome to.
So in terms of the pricing, Matt can answer that. In terms of the turnaround time, for about 40 to 50 tissues, the turnaround time is about 2 weeks. And this includes the showing you the images and also the quantitation data.
And in terms of pricing, our pricing is structured by tissue, and we have 3 buckets. The first level is imaging. So we have a first price for imaging. The second level would be basic quantification. Usually basic quantification doesn't include as much advanced data as we showed, and then there is the advanced quantification, which we do much more than what you saw today. I think what you have to keep in mind is that usually per data points per tissue, it will range between $250 up to $900 per tissue, and that's the range if you just do imaging to if you do everything on the data.
Thank you. Let's see. Has Genesis used this technology to analyze cancerous tumors in breast or other organs?
The answer is yes, and I'd be happy to take questions offline.
Good.
Stroma, yes, to make it short, yes. Imaging stroma and using our technique to look at fibrosis in the stroma area is of high interest. We have a clinical roadmap on breast, prostate, and skin. That might be a topic for a webinar later, but please give us a call if you want to learn what we do specifically in breast.
Great. And have fibroids due to endometriosis been analyzed by Genesis?
No. I don't think, we have not done it. That's a question that I can share with our partners in Singapore. They're a little bit more ahead of us in terms of oncology. We started, in April last year. We are focused on fibrosis, but I'd be able to answer your questions and follow up on that very, very specific question.
Well, lovely. I just really want to thank everyone. We have a few more minutes if you have any additional questions you'd like to enter. I so appreciate Dr. Shen and Dr. Petterjohn. It's a terrific presentation today. We will be recording. The session is being recorded, and it will be posted in a few days. We'll send out an email. And I just invite you, Dr. Shen and Dr. Petterjohn, if you'd like to make any final remarks. Is there a question that you wish someone had asked?
Leah, I'll let you go first.
No, I just want to say thank you for your attention and your time.
Yes, likewise. Thank you for listening to us. We believe we are bringing a very, very useful technique, and I'm looking forward to following up with you after the call if you have any questions. Our contact details are shown on the screen. And again, thank you so much, Joanne, and the GRC again for hosting and facilitating this session today.
Well, thank you. Everyone have a wonderful afternoon. Take care.